So we are going to move to the next speaker. Prisca Liberali is a senior group leader at the Friedrich Miesker Institute for Biomedical Research in Basel. So it's
recording in progress.
Also from Switzerland. She has been trained as a physical organic chemistry, and then she changed her field during her PhD to become a cell biologist. Her laboratory works on the role of cell organization in collective cell behavior and how these are coordinated by biomedical and mechanical cues. Her research has defined mechanisms dividing the formation of a 3D intestinal organoid, sometimes also called mini GATs, from stem cell cultures. In recent work, her group characterized the mechanism that triggers symmetric breaking during the formation of mouse intestinal organoid. She has awarded the Friedrich Miesker Prize in 2021 at the EMBO Gold Medal in 2022. Her talk today will focus on design principles of tissue organization. And Prisca, also, I will let you know when you have 5 minutes left on your time, okay? So don't get scared if I interrupt you.
Perfect. Do you hear me well? So today, thank you very much for the invitation. Do you see the presentation well?
Yes.
So today I changed a little bit and I decided to present some unpublished work we have on some also different type of organoids that we have been using in the lab. We, as was mentioned in the introduction, we work on intestinal organoids, and in the recent years we start working also on gastroloids. And so I will present now some of the work we have been doing in the past years on gastroloid development. That is our structure, 3D structure that recapitulate early embryogenesis and gastrulation. So what we are mainly interested in the lab is to understand how single cells cross biological scales. So how individual cells are able to form higher order structure in multicellular system. So this multicellular system have properties that are really at the higher scale, so tissue level scales. And so the interest is understanding how these individual cells are able to communicate and coordinate to make these patterns in vitro. The main challenge is that these tissues are not constructed machines. They're not built top down with individual cells that are put together one next to each other, but these are mainly built bottom up from short and long range interaction. And here you see you have individual cells that grow and divide. And while they grow and divide, they coordinate to really create new function at the tissue scale. So it's an individual cell that becomes and differentiate in a certain lineage. These are population of cells that self-renew and differentiate, creating a very complex organisms. And this is clearly genetically wired to make the process very precise and reproducible. Clearly, we have been seeing in the past years, both in vitro and in clearly self-intrinsic timers with oscillators with different type of transcription factor that have also create a rhythm. And this makes everything and the differentiation very precise and reproducible. However, it's clear that during development and during especially regeneration, the cells need to be able to sense the environment to take appropriate decisions. So every individual cells need to know where they are and so interpret the signals and of their environment to then take individual but coordinated decision. And part of this is really what we call the self-organization of the system and the space-time coordination that makes this process very robust and contextual in their behavior. So to understand this and this space-time coordination, it's clear that there are many biological scales involved, that you have the molecule, that you have the individual cells that are able to then sense the environment, create a population of cells that are coordinated, so both at the very short neighboring, but then you have population of cells of very long range, and all the way to tissue and organ. And clearly, individual cells need to transmit information forward to be able to interact with the other cells. And this you can have a different scales you can then have, and also different speeds. You have diffusion of morphogens is for sure much slower than the bioelectrical waves and span much less space. And so that was the question: how do these individual cells generate this emergent property at the highest scale? On the other side, there are the feedback and coordinated behavior to make the system robust. And this often is from higher scale back to the individual cells. And this is the part of the role of mechanics, of the extracellular matrix, and of a lot of different aspects that can be looked at. And especially we are interested in these feedbacks. So what are the feedback and the coordinated behaviors really to make the system? So in general, so that is what I think all the organoid field and this in vitro system really have been showing, that we can start from an individual cell. And in some time, depending on the system, you get to a full pattern tissue at the higher scale. So the main question is how an individual cell in an homogeneous cell population, so because at this point cells are actually cousins, in an homogeneous environment and in the same medium, manage to create a pattern tissue. So how do we create complex pattern tissue in vitro? Here you see an intestinal organoid, because this is most of the work we have been doing in the lab, growing from a single cell. It grows on the left, you have a maximum intensity projection of the whole organoids. In green, you have the plasma membrane. In red, you have the nuclei. On the right, you have us cut through the organoid. You see the cells proliferate. At the beginning, they have a very synchronous division, 2, 4, 8, 16, 32. There's interkinetic nuclear migration with the nuclei that are on the outside and then divide on the apical side here. And after an expansion of the volume of the lumen, you have the contraction and the formation of the crypt. So we are able to follow every individual cell for a very long period of time. And clearly to do this, what we were able to show is that intestinal organoid really recapitulate a regenerative response of the intestinal epithelium. So we start from the stem cells, or also other differentiated cells. We embed these organoids in matrigel. Then we grow them. And then we see that this formation of organoids doesn't depend on the stem cell marker LGR5, but on the YAP kinase that is involved in the Hippo pathway and is a mechanosensor and co-activator of transcription. And we see that the formation of organoid really requires this activation of YAP and an heterogeneous deactivation of YAP. And this heterogeneity of YAP is important to have the first notch delta lateral inhibition event. We have been also showing that these organoids are able to recapitulate the mechanical property of an intestinal epithelium, and they're able to form a shape, a proper shape. The reason we were interested in this system, again, is because there's this process that we call symmetry breaking, in the sense that we have an homogeneous population of cells, 16. They all come from an individual cell, so they're cousin cells, but actually only one become parent cell. And this is happening in every organoid system. You start from an homogeneous population, and then a subpopulation of them differentiate in a way, and then you can create this pattern. And how this can happen? So the way this happens is that, in a way, you have cell intrinsic mechanisms that sense the extrinsic cue to drive the collective behavior. You start from genetically identical cells, and the first emergent property at the tissue scale is a population of cells. And this population of cells will have intrinsic and extrinsic cell-to-cell variability. And this is not random, stochastic, completely random type of behaviors. You will have cells that are in G1 as G2. So in this population, you will have an heterogeneity in cell cycle phases. You will have different adhesion states. Some cells will have more neighbors. So some cells will be more spread. You will have different metabolic identities. And this gives individual cells in that population a different responsiveness to an homogeneous environment. That means that one cell that is in a specific cellular state is able to respond to an extracellular signal in a different way. And this will give an irreversible change in fate that then is a hotspot to drive the emergence of a collective behavior at the population scale. So this is really the point that we were looking at symmetry breaking. We did really quite some advance in the intestinal organoids, but recently, like a few years ago, a PhD student in the lab, Marietta Zingler, started to work on gastroloids. So just to give you a bit of an introduction on gastroloid, these were published. I'll give you started from Alfonso Martinez Arias, and then recently also by different other people. But what is this system doing? So this is recapitulating early embryonic development. There is the symmetry breaking. There is the formation of the three major body axes. There's germ layer specification and acceleration. We have the expression of multiple cell type markers, also very late. There's a correct temporal and sequential expression of the genes. So they're not only expressed on the right place, but especially on the right time over the formation of the gastroloid. They can recapitulate heart and gut development. And if re-embedded later on in matrigel, also have somites and trunk-like structures. And to form this gastroloid, we start from 300 mouse ES cells. We aggregate them, we let them grow in this like hanging wells, and then between 48 and 72 hours, there's a pulse of Wnt activation. This pulse of Wnt activation that is done here then drive the initial part of the morphological symmetry breaking with the elongation and the formation at 5 days of these structures that have a full anterior-posterior axis. There's some limitation. There's no really rostral identity and neural derivate. So it seems in the normal protocol, there's not real neural tube or neural crest. There's no extraembryonic tissue, but there's no clear internal organization of cells. So there's not a lumen or like clear organization of the cells. And this can be done both by mouse, human, and zebrafish embryos. By imaging, this is how they look like, because we combine both imaging and sequencing time course. Here you see at 24 hours, you have the formation of the structure. With the Wnt activation, we start expressing brachyury. And this gives then later on the difference between an anterior-posterior axis with the brachyury expression and the acceleration. So to first look at these development of these gastroloids, we perform a time course of single-cell RNA-seq. Here you see the first time course of gastroloid development. Here, you see the different clusters being annotated. We have an initial population, 12, that are the naive pluripotent cells that are really the early stage. And then over time, there are the differentiation of the cells that goes through an epiblast type of structure here, a very big homogeneous population. And later on, here at this point is more or less the Wnt pulse. You have the differentiation on all the different cell types. Clearly, in this protocol, the majority of the cells that you see, 14, 15, 16, these are a lot of mesoderm derivate. And then you have a primitive streak-like type of population that goes from this epiblast to these differentiated cells. At 10 here, you see there's also gut differentiation or the NMP, so the neural mesodermal progenitor. Interesting, and I'll just highlight it here, and I'll go back. There's also this population of ectopic pluripotency, and I'll go back to it a bit later. To then color code these over time, you see that the cells are, sorry for the resolution, it's just a little weird. So the SARS start at 0 hours, all as naive cells. They start progressing out of the naive states, and so at 24 hours, they move out. Between 36 and 48 hours, that this is, and 52, so really initial, but you see the dark blue and the purple are the first 2 time points. So the cells are in the epiblast state, and then what happened is that with 52, the cells start moving and moving down. So here you see in green, 56, 60, and 72. So this is after the Wnt pulse, and the cells start on one side moving down here to the differentiation and the expression of here, but then there's a subpopulation of cells also moving down to this ectopic pluripotent. This population you see is also stable at later time points. So this seems to be a population that is formed after the wind pulse from an epiblast population, while the other one really goes just to the differentiated state. We start comparing some of the cell type over time. Here you see the NMPs, and you see we compare them between the NMPs in the gastroloid and the NMPs in the embryo, and you see that there is a very good correlation between the three, both the NMPs, the presomatic mesoderm, and the gut. So we are managing to create cell types at later time point that really look like in vivo. There's then an ectopic pluripotency. Here, this is exactly what you see. This is the SOX2 population. They grow here, but then they maintain this population over time. And they also have really naive markers like ZFP42. We have been looking a bit more and deep in this population and seeing what they would really represent. And it seems that some of them might become more like PGC, so PGC-like cells. Some others really start changing expression profile over time. So what we think is that this population is actually almost a bit of a in vitro artifact, because these would be cells that in vivo would not see wind. And so they almost revert back to a naive state. But depending on the environment where they are, they can slowly differentiate in other cell types. So to finalize the single cell RNA-seq, what we also did is that we took these cells from the gastroloid and we embedded them with the embryo data set. So this is the data set of John Marioni's lab that went from E6.5 to E8.5. And you can see in the left, the cells from the gastroloid are color-coded, while on the right, only the embryonic cells. You see at later stages, the cells, so after 84, after wind, the cells are very similar between the embryo and the gastroloid. However, if you see how they get there, this is like 0, 24, and then at this point you have the wind pulse and you make the different cell type. While the embryo really goes more through a gastrulation type of structure. And probably the differences also of this behavior is that the cells are also differently spatially organized. So after looking at what are the different cell type and how they emerge over time, we wanted to look at the spatial organization of the cells. So to then perform this experiment, what we did, we had the time course and fixing at every day from 24 to 96 hours. And then we performed some imaging. We looked at around 7,000 gastroloid. We had 22 antibodies, and then that were staying in parallel. From those, we were then segmenting every gastroloid and create 100 segments over time. And so in a way, it was to do like a patterning of the gastroloid, but computationally on the images. Then we had an inside-out type of features and the superpixel. What we also did is then to create a trajectory in pseudotime of this gastroloid. So you can imagine that if you fix at a certain time point, not all the gastroloid will look exactly the same. And so you have this variability because of the different pace of progression. What we do in a way is using these features, especially the size and the number of nuclei, to then align the gastroloid over time and look at the molecular progression during their differentiation and development. This is how the gastroloid look like. You start from the cells. And interestingly, what we see is that this SOX2 that actually is a marker of this early population of naive cells is very central in the core. When then we give Wnt at 48 hours, we see that the response of brachyury is that only the cells not expressing SOX2 will express brachyury. And then you have this binary response with the SOX2 pole and the brachyury differentiation. We also then looked more a bit at some of the data. And what we saw is that there's a core fibronectin, here you see it in red on the top, that actually maintains and is present much earlier than the SOX2. So then you have, in a way, a first fibronectin core in the gastroloid that then correlates very strongly with this core of SOX2, and then is maintained over time and correlates with SOX2. So at this point, what we think is that we start from an homogeneous population of SOX2 cells. There's an heterogeneity in the extracellular matrix with the core of fibronectin that then create this heterogeneity in SOX2. So when we get at 48 hours, we have this heterogeneity in SOX2 with the core cells have much higher SOX2 and the outside cells that have much lower. When we did the Wnt activation, what is actually happening is the outside cells are competent for differentiation and acquire brachyury, while the core cells revert to these naive pluripotency or naive or ectopic pluripotency, how we to call it. And some of them over time then also express some PGC. I think this is quite interesting, and it was interesting for us to see. And so first we wanted a bit to play with the protocol to see if we could validate some of these responses. And so the first thing we did is to activate Wnt a bit earlier. Activating Wnt earlier would mean, as you can see here, there's much higher SOX2 over time. And then when we get brachyury, there's much less brachyury at the beginning of the system. And over time, we get really weird gastroloid with actually much more multiple foci. As you can see here, in red, you have 1, 2, 3 foci of brachyury. So what we think is in a way that this correct proportion between SOX2 high and SOX2 low before the activation of Wnt is very important to create the good number of cells required to create a gastroloid. The other ways to change this heterogeneity was to change the number of cells. In the field, it's clear that the initial number of cells for gastroloid development is very important. And it's this center column with 300 cells. And so we start making gastroloid with 150 cells or 500 cells. Here we have a different fibronectin core and a different SOX2 core that then gives later on a different brachyury response. And again, also here, if you have way too much SOX2, you have this multifoci interval. So what then we think is that this SOX2 variability and a good number of SOX2 high, SOX2 low gives the system a very important responsiveness to the system, where then the Wnt activation is then giving different behavior to the different cells, because the cells are differently competent. So even being in the same environment and the same medium, some cells will respond differently. So the cells will differentiate, and then one goes back in a more naive state. Some of them will then differentiate again, while the others continue the trajectory of differentiation. To look at the system then even more, so we thought, okay, we have 3 main places where the gastroloid development is happening. One is this first SOX2 variability. Second is a binary fate choice with activation, non-activation. And third, there is the elongation process of the gastroloid. We believe this is a very mechanically oriented, because the two, let's say the two fates, the core and the non-core, are mechanically very different. And so there's just an opening and then an elongation of the system. So to understand molecularly how this works, what we did is to do a phenotypic compound screen. And this screen was not only, it was done fixing at an endpoint, so at day 5, it's 120 hours, but we had 3 different regimes at which we would add the compounds. One was adding the compounds between 32 and 72 hours, so covering the before the wind pulse. Then after that, there was the in between, so during the wind pulse, and then the second one is after the wind pulse to look more at the elongation. To do this, what we did is to use a screening library of 84 compounds, 68 unique targets, and we used 40 gastroloids for every condition. This is the library of compounds that we use. These are very strongly enriched in signaling pathways involved both in development and regeneration. With this, we imaged around 9,000 gastroloid for almost 2 million images. We stained for the 3 germ layers, so ecadherin, brachyury, and SOX1, so endoderm, mesoderm, and ectoderm. And then what we did is first trying to extract a lot of feature at the full gastroloid level, as I mentioned before. And then the other one was to cluster these features to look at the phenotypic and what we call the phenotypic landscape. So that actually means how many phenotype can you make? Actually, we have this complex system. We have 300 starting cells. We probably have around 800 cells when we add wind. But actually, how many phenotype can we make? And this is what we call the phenotypic landscape. You see here on the left, we have the first 2 gastroloid, the wild type gastroloids with the different, and as you can see here from the different stainings. Then we have the SOX1 phenotypes. This is completely SOX1 positive. This you have SOX1 with the ecadherin. Here you have pretty good one with the SOX1 more anterior and the brachyury more posterior. Then you have the SOX1 with a big ecadherin, in a way is the mirror image of this one. Then this is brachyury with ecadherin, and these are more brachyury phenotype with a lot of brachyury. So that was already interesting. You cannot make actually anything. In a way, you always make some ecadherin, you always make some mesoderm apart 4 and 5 and 6. So I think that this is quite interesting. But the other part is like, can we start stratifying this depending on the time point? Because I don't know what happened to this presentation and the resolution. I'm really sorry. So what happened here is that we have this UMAP, and then you see these fuzzy dots, and all these dots are one gastroloid, and they're color-coded by their phenotype, and they're differently stratified by the different time points. So here's a UMAP of all gastroloid from early time point for the wind pulse and for the elongation. What is quite clear and striking from the beginning is that you have this subpopulation of gastroloid that only comes up if we perturb before the wind pulse, not during and not after. And this is quite striking, is these ecadherin with a point of SOX1. In parallel, over time, what you see is that there is an increase of SOX1. So actually, to increase a more anterior part of the gastroloid, we need to perturb later on. So we start then looking at the different genes and how they perturb the different organoid, the gastroloid, and then you can see here, this is color-coded by the phenotype, 32, 48, and 72. Clearly, we have this cluster of genes with FGF, MAP kinase, and some calcium calmodulin kinase that have this exactly what I showed you. They start, when perturbed before the wind pulse, to be this ecadherin ball with a SOX1 part, while later on they become only SOX1. The other part that was very interesting is these wind phenotype that at later time point give a quite strong SOX1 phenotype, but very different before. So then we went a bit deeper and tried to validate some of these results. And so initially we looked at this MAP kinase and FGF requirement for gastroloid progression. And so in this case, what we did, We didn't fix at the end time point of 120 hours, but then we fixed at earlier time point to see how the gastroloid looked like when we gave the Wnt, because this is clearly a phenotype that comes up only perturbing before Wnt activation. Here is how the experiment looked like, and I will guide you through both the images and the quantification. So here you see at 48 hours, you have the DMA salt, you have the core of SOX2 and fibronectin. When you activate with brachyury, you get the outside and the inner and the outside. And then later time point, you get this brachyury here in the posterior part, more in the anterior. You have this DPPA4 that is a marker for this ectopic pluripotency population. And this you can then see the quantification over time, 48, 72, and 124 of the different. Then what we did is then to perturb with the FGF and the MAP2K1 inhibitor. And indeed, you see that at later time point, we have a very strong accumulation of only DPPA4. And this DPPA4 is a marker for naive especially for the pluripotency. And here you see that already at earlier time point, we have much higher SOX2. So this SOX2 population is staying much more homogeneous at 48 hours. So when we give the Wnt, we have much less brachyury expression, and we then have this very strong population of DPPA4 that also express cadherin. So this, we characterize these very early stages. But the other things we wanted to do is, can we then use what we learned by both the mechanism of formation of the gastroloid and of the screen to make better gastroloids? One of the limitations is not having this anterior part. And so, as I pointed out in some of the previous slide, is that we have this early SOX1 population, this very clear one, that's quite strongly upregulated in the beta-catenin and a porcupine, so it's an ADOPP2, so it's a Wnt inhibitor, a Wnt secretion inhibitor. And so the idea here is, when perturbing during the pulse or just after the pulse, push these pluripotency core and ectopic pluripotency to a more anterior and rostral identity. And this we are doing with the inhibition of it. And indeed, when you see here 96, 120, and 144, so we even have been pushing them even further, when we inhibit with beta-catenin or GSK, we have a much more anterior SOX1 population, as you can see it here. So this is really work in progress, and we are trying to finalize some of these experiments to really stain this SOX1 population with other more anterior marker to see if this is more neurotube-like, if this is more really rostral. So I think that would be really interesting to understand how much can we push a full developed gastroloid. And so with this, I'm getting closer to the end of my talk. And then so to summarize, what I showed you is that the formation of gastroloid goes in these, let's say, 96 to 120 hours, starting from an homogeneous population of ES cells. Very early on at 24 hours, we start having this core of fibronectin. Then, depending also on signaling of MAP kinase and FGF, and also TGF-beta, there is an establishment of SOX2 cell-to-cell variability that is spatially organized with a core of SOX2 and an outside of non-SOX2 cells. This heterogeneity that is actually reversible and it's not even huge, so you have this cell-to-cell variability of SOX2, still gives the cells a different responsiveness to Wnt. And so when we give a Wnt activation, there's this binary phase choice. Then there's some endogenous Wnt signaling activation inside the gastroloid that does not allow to give the full maturation. And so with Wnt inhibitor, we are then able to even push further the elongation and the anterior and posterior axis of these gastroloids. And what we believe is that these ES cells sense their spatial organization, so sense very well where they are, and adhesion code probably, to regulate their competence for Wnt activation. So that would mean that also in vivo, when you have these different cells, clearly they're gradients, the organizer, their signaling organizer, there's the extra embryonic tissue that differentially express different patterns. But there's also some intrinsic measurement of the organization and adhesion state to then react differently to the Wnt. What we see, and we did an ataxic of this process, is that probably at 48 hours, there are like some enhancer of ES cells that are still open in the core population. And so when they see Wnt, these cells that have the enhancer open are able to revert back to a more naive state.
Prisca, we need to wrap up. So,
yeah, it's the last slide. And so with this, we are now moving on more looking at the dynamics of the gastruloid formation. And so here we are developing this microscope to look at the dynamics of gastruloid formation and try to then also look at the dynamics of these different cells. And with this, I would like to thank the full lab, especially Simon Suppinger and Marietta Zinner for all their work into the gastruloid development, the collaborators, and all of you for your attention, and I'm happy to take questions.
Thank you very much. That was an excellent talk. So now we have some time for some questions. Is any question in the audience?
Hello, this is Abhishek. Talking about the gastruloids, if we consider the normal development process, there are organizers present into the embryo, which guides or directs other cells and orchestrates their cell migration as well as their spatial arrangement into the embryo. So do you observe any such organizer-like structures during gastruloid formation?
Yes, so we looked for them. We don't see a very stereotypic formation of organizer, but we still see very strong migration after the Wnt pulse. So I think initially is during gastruloid development is that then when the different cell types form, there's like a migration and a sorting out of the cells to then that recognize each other. And then probably later on, they start making some organizer, but initially is more a sorting out process. And then there will be some organizer formation.
I see other person over here.
Wonderful talk, Prisca. Thank you very much for this talk. I just wanted to know your experience. Do these time frames of these initial few hours or the first 4 days or so match between the mouse gastruloid formation and the human gastruloid formation? What is your experience?
The problem and I think the main issue is that a lot depends on how you culture your ES cells and the ground state where these ES cells are. And the ES cells, the mouse and the human, are in very different states.
Yes. Yes, that's right.
And so I think that is one of the biggest differences of the two protocols and why they are not looking very similar. The other aspect is that these gastruloid formation don't have any shape of any form. And so what we are probably looking at is actually mechanism of differentiation of the different cells.
Probably in vivo,
So
there's a shape difference between the way the implantation, at the time of implantation, the embryo looks between the murine version versus the human version. There's a disc formed in the case of the humans, whereas in the case of the murine, it's a totally different elongated shape, yeah.
Yeah, so this is exactly what we think, and one of the reasons we personally don't love the gastruloids. I think they're a very good system for doing a lot of things, especially to make end time point cell types. Okay. So I think it's still the best in vitro system to make NMPs, to make mesoderm and prismatic mesoderm, to make some gut cells, because all the other in vitro early embryonic system don't get that far.
Yeah.
That said, I think they're not really called for what they do. In the sense, they don't gastrulate.
Okay, okay, okay.
Is not so. I think it's a system that makes all the cell types that then, when they are made, they migrate to each other and they start creating structure. They start creating organizer. Before actually the wind pulse, so you have all the cell type you differentiate, and then the wind pulse create these cells and this heterogeneity of cells.
Yeah.
But they don't go through any morphological changes like gastrulation. And so I'm trying to be, it's like, so I'm not sure exactly what we are recapitulating in these early stages. I think there's a lot of in vitro system. And so a full comparison between human and mouse might not be the best system to look at for the first 4 days.
Okay, thank you. Probably if you have a trophectoderm cell association, maybe they will progress to a more in vivo kind of condition.
Yes, so there was some work from Matthias Lutolf that really looked at that, and there was a paper where they added some other cells, and then they would have the wind secretion that would be local and not homogeneous, and that would already improve a lot, especially for the formation of more an anterior part. Because especially in vivo, not all the cells see wind at that time.
Yes.
Some cells don't in the anterior. So I think that that's having some ways to make better gastroloid, and so using this data also to enable us to make better gastroloid would be great, I think.
Okay, we have time for one more question. I see one hand here.
A very nice talk. This is regarding the role of ECM components in cell-fate decision. So you showed nicely that the fibronectin, the SOX2 positive cells are fibronectin enriched, right? So is it, does it make sense that you can really, if you want to, can manipulate the cell fates by culturing them in different conditions? Like, have you tried putting fibronectin or injecting in the other pole and see if that changes the cell fate? So I was just curious.
Yeah, so we did that. We both removed fibronectin with some sRNAs. We added some fibronectin. We also added some RGB peptides, and we see a big difference in the behavior. That said, the fibronectin effect doesn't happen in every cell lines, because so we did this experiments in 6 or 7 different ES cell lines to see what is, what are the different behaviors. And there we clearly see that there's always this heterogeneity in SOX2, but there is not always the fibronectin core. So I would imagine that depending on how you culture them and depending a bit on the different cells, there might be different involvement of the role of ECM. Clearly, when we remove DCM in these cell lines, we don't have this, we don't keep this pluripotency very long, and the gastroloid don't make it. So I think that would be very interesting longer term. We didn't do it systematically. We didn't try different ECM with different cells. I think would be very interesting to look at the role of the DCM for that.
We have time for one more short question.
Yes, can I speak?
Okay.
Okay.
So only one more.
So very nice talk. So I'm interested in this 48 hours wind pulse. So you showed that beta-catenin is upregulated. So you're saying that it is a canonical wind ligands, they are basically enriched at 48 hours?
Wait a second, give me a second. Give me a second because my window is open and I don't hear well. Sorry. Okay, the Icadiran population?
No,
Sorry.
I was talking about wind pulse. So at 48 hours, when you see this beta-catenin, so you are saying that the canonical Wnt ligands, they basically are upregulated at 48 hours because that is fueling beta-catenin or something else?
So the problem of the Wnt ligand, the endogenous one, we cannot see it with a single cell RNA-seq.
Okay.
And that is the major problem. We know that there is some endogenous Wnt that is expressed, because if we don't give the wind pulse, we still see some elongation. So there is some endogenous, and then we see an effect if we then inhibit it.
So have you seen the Wnt receptors on these cells? Like Frizzles, LRP56, ROR?
Yes. But the problem is that, again, we don't manage to see it well with the imaging. So I think that would be really great to see which cells have it or not. The point that we know is that they all respond to wind, because if we do a TCF line, GFP, they all turn it on.
But you know, beta-catenin can be stabilized without wind also. Beta-catenin can be stabilized without wind signaling also.
Yeah, and so this is why we have been, especially the last part, you see, we both inhibit wind and beta-catenin. And this is the reason, because we have the feeling that they might be not always the same way in the different steps. And so combining different ways of inhibiting wind, because this is exactly, in some of the data we had, it was not clear what was the response. They all respond to wind very clearly at 48 hours, because we did all the control, because at a certain point we were not sure if maybe the core cells would then see less wind. But on the exact response and what's the role of wind, especially during the elongation, I think it's really not clear. So the problem is with the single-cell RNA-seq, you don't see it. They're all too low expressed. And to have good stainings of all of these is not trivial. So this is a work in progress, and we are trying to have different compounds that then activate and inhibits the pathway at different heights.
Thank you. Well, we don't have more time, unfortunately, sorry. But let's thanks one more time, Dr. Liberali, for this amazing presentation. Thank you very much.
Thank you, and sorry for not being there. That's been great.
We miss you.
Bye.
Bye.